Thank you very much for the introduction. Thank you for being here. We are a Swiss company, as you can see. We're based in the Biopole near Lausanne. And we are fully focused on the development of allergy vaccines or allergy immunotherapy. Our goal is to develop what we call ultra-fast allergy immunotherapy. This does not exist today. And the way to visualize it is pretty simple. We're trying to achieve a long-lasting protection against allergy symptoms with only 5 injections before the allergy season. And you will see we already have some data going in that direction. And this is to compare with the subcutaneous or sublingual treatments that are currently on the market. And those treatments require 3 years, sometimes even more than 3 years. So these treatments are associated with huge compliance problems. More than 50% of patients are no longer on treatment after 1 year. And up to 80% of patients drop from treatment before the 3 years. So the medical need, the room for improvement is definitely there. It's for us to demonstrate that we can do what we are up to. The market, you can imagine, can be very large. We're talking about a pool of about 500 million allergic patients. These patients are mostly covered today by symptomatic treatments, as you know, antihistamines, and the allergy immunotherapy, the 3-year allergy immunotherapy, represents about 10% of the market today. We think that if we can bring these ultra-fast treatments, and we've done proper market research internationally, we can definitely generate, first completely change the market and generate very significant sales. You have some estimates that we have produced here within Anergis for the 3 allergies that we're after to start with, at least. House dust mite allergy, ragweed pollen, and birch pollen allergy. The technology we are using is to desensitize allergic patients, using peptides, which are derived, which look like the allergens in a way. We're using peptides reproducing the sequence of the major allergens. And we administer those peptides. Now, this has several advantages. We disrupt the 3-dimensional structure of the allergen, obviously, by using separate sequences, separate molecules of peptides. And this gives us the absence of IgE binding. So when we administer our peptides to allergic patients, they do not respond with an allergic reaction. And this is what allows us to increase the dose, and we think the cumulative dose is what matters for the long-term effect. And in our first studies, we gave as much allergen equivalent in 5 injections as with the standard treatments over 3 years. So you could call it massive doses. This was done safely because there is no binding to IgE. The fact that we have long peptides maintains the immunogenicity also of the peptides, and because the long peptides, unlike smaller ones, are internalized by antigen-presenting cells and activate a T cell and B cell response very efficiently. And last but not least, we have synthetic peptides, therefore they're high purity GMP according to current pharmaceutical standards, which was not the case with this field, where natural extracts were used most of the time. Now, with the first generation of our COP allergy vaccines, as we call them, our allergy immunotherapy, we have shown some very interesting data in phase 2b, where we demonstrated that the product is efficacious. So when the peptides are combined with aluminum hydroxide, the product is efficacious during the first season following treatment, but also during the 2nd year without repeating treatment. We are the only company able to show this type of data. We have also shown on the right panel that the antibody response, the specific IgG4, which are marker of immunotherapy or successful immunotherapy, are still markedly elevated 4 years after the 5 injections. without any repeated treatment again. So this is very promising and was very promising until we came to this last phase 2b study where we conducted pretty much it was like a phase 3, it's a registration trial with 3 groups, placebo, 50 microgram, which we think is our main dose, and 10 microgram because we wanted to show that it's less efficacious and that we can go to phase 3 with the 50. And what we found in this study, well, first let me say that we used in that trial more sensitive, more allergic patients. We selected more severe patients on purpose because we wanted to show a bigger effect or larger effect of the product. Well, it turns out we got a smaller effect. What we showed here, it's a bit complex, but the first line here is the primary endpoint. We showed a highly significant, we met the primary endpoint with a 7% improvement versus placebo. So it's nice to meet the primary endpoint, but it's not enough to provide a proper benefit to patients. And regulators would never accept a product like this. We also showed in the same, and we think it's related with the limited efficacy, sensitization to the peptides, which developed over time during the treatment period. So with this, we concluded that this product, this first generation, cannot be developed further. We moved into looking at what could we do to improve the product, and we decided to modify the adjuvant. We know aluminum hydroxide is promoting Th2 response and therefore sensitization. This is where we came close to Mymetics, who is in the room and presented very before me. We went into this collaboration with Mymetics because what we are looking for is a Th1 promoting formulation in which we can include a TLR agonist in order to boost immunogenicity and prevent the sensitization to the peptides. We have entered into an option to license, so we know where we're going as long as the results of the current mice study gives us what we expect. And what we expect is shown on this slide. We expect to see a higher Th1 than Th2 response. We expect to see no sensitization to peptides, and of course no clinical signs that would justify or not justify going forward. And if everything goes well, we will know by the end of the year. If everything goes well, then we're going into the next step. And this is why we are presenting at this meeting. The next step is to redevelop this second generation containing virosomes with the best of the different options we are testing in the mice study, because we have included several options of virosome peptide combinations. And at that time, we want to raise 13 million, so that is early 2019, in order to advance together with Mymetics the development of the new formulation. We have to redo a tox, it's a new product, and then we will go into phase 2A, and it's really in phase 2A by mid-2021 if everything goes well. It's with this phase 2A that we will prove that the second generation is better than the first in man. And by the end of this year, I'm hoping we will have shown that in mice, it is what we find. The team is here. We have a management team who's been around. We have accumulated quite some experience, as you can imagine, in the field. And this is very useful. I'm just showing one man. He's the founder of the company, François Spertini from the University of Lausanne. To conclude, this is a summary of the investment case, because we are here for raising again 13 million early next year. We have a proof of concept from the first generation, and we have data that are truly unique. But the first generation was not good enough. We have a very good go at a second generation that could be much better, and hopefully we'll show it. And then we can proceed. Our main goal in the coming years is going to be to show in man that the second generation is better than the first. Thank you very much for your attention.